Yes, why chromosomes, the number of chromosomes is reduced? Why there is a formation of haploid cells? Why there is no, why not the same number of the chromosomes are shifted to the next cells like mitosis? So these are the importance. Like number one, that there is the maintenance of the chromosomes number generation after generation, that how the number will be remain same from one generation to the next generation. So first of all, we know, we will take an example of human being, and we know that humans have 23 sets of homologous chromosomes, mean 46. Total number of chromosomes is 46, and when we will make the pair, then there will be 23 pair, and each homologous set is made up of two homologous, and you can see that there are 23 homologous chromosomes, and that one is an example of human cell. So we know that in the fertilization, that process is very important. We know that what's the meaning of fertilization. There is the fusion of sperm and egg, and we know that sperms come from the male gamete and egg come from the female gamete, and when these cells will be fused to each other, then there will be formation of zygote, and zygote is a fertilized egg where there will be formation of an embryo, there will be developmental stages, and then there will be formation of an individual. In the mitosis, we know that after fertilization, then mitosis will be started for the development and for the growth of an individual, right? So after that process will be started, mitosis. But before this, before going to fuse, think about it, that if number will remain same, suppose like 46 from the male gamete, 46 from the female gamete, if number will not be reduced, then what will happen? 46 plus 46. So what will be the next number? It will be 92, mean the number will not remain same. When number will not remain same, then how the information will pass over to the next generation? We know that we have genes, and there are different codes for all the information. A single cell carries all the information of your body. So if number will be changed, then we will not able to maintain the number of chromosomes. So through this process, the reduction is very, very important. First of all, in the male gamete, there will be reduction of the number of chromosomes, and the female gamete, that is also very important for the reduction of the number of chromosomes. When number will be halved, then you can see in this diagram, 23 from a female gamete, 23 chromosomes are present in egg cell and 23 chromosomes are present in sperm cell. When both will be fused, then the total number will be restored. Clear, girls? Because all the species have their specific set of chromosomes. They have a specific code through which the information are passed from one generation to the next generation, right? So it's very, very important to maintain the number of chromosomes generation after generation. So this one is an example of human cell, and now we will take an example plants, that how plants, because plants are also living organisms, and how plants maintain their number of chromosomes generation after generation. In plants, the process is slightly different, because there are two types of generation, sporophyte and gametophyte generation. Sporophyte, please focus on this diagram, then you can easily understand what I am saying exactly. That sporophyte produces the spores, through which process meiosis, mean the number will be reduced at this stage. Clear? And then those spores will give rise gametophyte generation. And gametophyte generation, because there will be two types of gamete, male gamete and female gamete, and they have the haploid number, because the number is already reduced when there is the formation of the spores. So after the fusion, what will happen? When there will be zygote formation, the number will be restored, and zygote basically again give rise to sporophyte generation. For the sporophyte, you can think about that there is that one is the large in size, like tree. Think about of a tree which consists of root, stem, shoot, branches. So here there will be the gametophyte generation. So move toward the flower part. In the flower, there are reproductive part. Inside the flower, there are male and female part of the plant body. Inside these parts, there will be formation of male gamete. In the male part, there will be formation of male gamete, and in the female part, there will be formation of female gamete, and after that there will be pollination. Pollination means transfer of pollen grains, transfer of sperms from the male part to the female part, and then there will be fusion, and fusion means fertilization. Then there will be formation of zygote, and you can see that in this whole diagram that how the number will remain the same and how the same number will be transferred to the next generation. So it's very important to maintain the number of chromosomes generation after generation. Next significance, that there is a variation, production of variations in next generation. It's very important. The chromosomes pair of each parent undergo crossing over. So crossing over is important for the formation of variation. Then there is the fertilization, because if we will take an example of sperm, 1 of million sperms fuse with one egg. So it is also responsible for the variation, and beneficial variation helps organisms to adapt to the changes in environment, right? So that one is also important for the variation in next generation. So what will happen if there will be any error? We have discussed about the separation of chromosomes, separation of chromatids. Normal separation of chromosomes and chromatids is known as disjunction. And if there will be no normal separation, then it will lead toward a problem like mitosis. In mitosis, we have discussed about the cancer. And in this case, if there will be no normal separation, then what will happen? You can see this diagram. If there will be no normal separation, then in one cell there will be decrease in the number of chromosomes, and in the second there will be extra number of the chromosomes. It will lead toward the trisomy or monosomy, mean that maybe the individual have 45 or 57, because there will be formation of abnormal gamete, and when abnormal gamete will fuse to each other, then there will be formation of abnormal individual. Like there will be maybe there will be decrease in the number of chromosomes, and maybe there will be an extra number of chromosomes. So these are the problems which will be arised if there will be no normal separation of chromosomes, right? Now I will recap the topic meiosis by comparing with mitosis. And you have to focus in this diagram. You can see that there is a simple cell, like where there are the chromosomes are present in the form of loose thread-like structure, which is known as chromatin, and black dot is showing nucleolus. And in the next stages, what will happen in mitosis? There will be the condensation of chromosomes, formation of spindle fibers, and the same thing will be occurred in the meiosis prophase 1, but there is a difference that in mitosis there is no pairing of homologous chromosomes, and in meiosis 1 you can see that there is a pairing of homologous chromosomes. They will be crossing over. In the next stage, in the metaphase, you can see that in the metaphase of mitosis there will be alignment of single chromosomes, but in meiosis metaphase 1 there will be alignment of homologous chromosomes. And in the next in anaphase of mitosis there will be separation of sister chromatids, but in anaphase 1 of meiosis there will be separation of homologous chromosomes, means separation of whole chromosomes with sister chromatids. And at the end, in the telophase, in the daughter cells, you will see that in the mitosis there will be the same number of chromosomes. At the start you can see that there is a 4 number, at the end the number will remain same, right? Diploid number of chromosomes. Each chromosome has single chromatids, right? But in meiosis 1, at the end you will see that there will be the reduction in the number of chromosomes. Now each cell will have number of chromosomes is 2. At the start there is the number 4, but at the end each cell will have the number of chromosomes 2. So now the daughter cells, daughter nuclei contain haploid number of chromosomes, and each chromosome has 2 chromatids, right? So I'm sure the topic will be clear. right? You have to go through this topic mitosis. If you will learn the topic mitosis, then you can easily learn the topic meiosis. If mitosis will not be clear, then there will be a difficulty in understanding the concept of meiosis. So please learn this topic with the help of diagrams, compare the diagrams and make the points, that what are the points. Like you can see that in the prophase, what we have discussed there is a condensation of chromosomes, there is a degradation of nuclear membrane and nucleolus. We have discussed there is a formation of spindle fiber by joining tubulin protein. There is a formation of kinetochore spindle fibers, right? And in the next stage, we have discussed there is the alignment of chromosomes. In anaphase we discussed separation, right? And in the telophase, which is the reversal of prophase, 70 to 80% topic is similar. Just the difference is this, that there is a pairing of chromosomes and there is no pairing of chromosomes, right? Process is same. Like if there is an alignment in mitosis, the same process is going on in meiosis, but the difference is just because of the pairing and no pairing. Homologous pair is aligned or not, or single chromosomes. Is that clear? Okay.